Today's review is of The Codebreaker, Jennifer Doudna, Gene Editing and the Future of the Human Race by Walter Isaacson. Our presenter today is Dr. Mitch O'Connell, Assistant Professor in the Department of Biochemistry and Biophysics in the Center for RNA Biology at the University of Rochester's Medical Center. Dr. O'Connell is a research scientist who works on understanding how CRISPR-Cas systems function and how they can be used to treat genetic diseases. And he previously worked for Jennifer Doudna. Please join me in welcoming Dr. O'Connell to our podium.
Just because I move around a little bit, I'm going to use this one. So thanks very much for the gracious invitation, and a pleasure to be here today. I'm hoping I can distill what's quite a technical book down into something that you can take away today. I don't know how many of you have read it yet. It's a good portion. And so I will start with my conflict of interest. That I used to work for Jennifer for 4 years. It was a wonderful time. This is when I was at Berkeley, California. And so I did some really interesting work with Jennifer, and then as part of that, I was able to secure a faculty position at U of R. So she's been instrumental in my career, and in, as you can see, many others as well. And we got really excited last year, because I'm just going to jump to the chase, that Jennifer and Emmanuel Charpentier were winners of the Nobel Prize for what was talked about in this book. And because of COVID-19, we weren't able to celebrate all in person, but due to the powers of technology, we were able to actually have a big Zoom session with all the alumni from the lab over the last 20-odd years. And so this was really a culmination of the excitement that has kind of been captured in this book. And something to highlight, and I don't know how well everyone can see the screen, I keep bumping this, is that only out of the 184 recipients of the Nobel Prize in Chemistry, only 7 women, and we're going to talk about 2 of those today. And so that kind of really sets up the story about Jennifer's early life and how her kind of dogged curiosity led her to make some remarkable discoveries. And so this is all really well captured by Walter Isaacson. He's written a lot of really wonderful books before, and is quite esteemed as not only an author and a journalist, but also a professor of scientific history. To be honest, I haven't read many of his other books, but I feel like what he captured in this and Steve Jobs' book was really quite impressive. And so I suggest you all read the book. I really enjoyed it. And so the way I kind of see that how the book was kind of dissected, at least in my mind, was really setting the scene of... Is this really bothering everyone? I think I have this. Keeps hitting my glasses. Sorry. Is really setting up the scene of a young woman that fascinated by nature early on, and some of the small barriers that it took to overcome to become the powerful scientist she was today. And so growing up in Hawaii, feeling like a bit of an outsider, having her school counselors say science isn't for women, and really setting up that initial tension that's kind of carried out through the book. And then it kind of switches to understanding some of the basic science, from the composition and structure of DNA, genetic code, to understanding DNA's cousin, RNA, which didn't really hit the limelight until much more recently with the COVID-19 vaccines, for example. And then really diving into not only the initial hypotheses around CRISPR, but also the applications and the gene editing. And that whole framework is kind of mingled in with the sometimes the competitive and sometimes curious personalities of the scientists involved. And I think that's what this book did a really good job of, is saying that science is done by humans. Humans are fallible, humans are competitive, selfish, but together they work together as a consensus to really build something powerful and have at the forefront really the goal to help the human race. And then the book kind of switches towards the end, looking at what happened during COVID. And I don't go into much detail about some of the newer advances there because I thought it was a little tangential. But also the promise and peril of using this technology to target diseases, and where do you draw the line for something that's so powerful? And so really a lot of the bioethical aspects of the technology. And so the book really starts, and Walter really does a great job of setting up the story, particularly with this girls don't do science kind of mentality that came out early. And I think what I really liked from this is a lot of people have maybe heard of the double helix and Watson and Crick and Rosalind Franklin, and this idea that they solved this beautiful structure of the double-stranded DNA double helix. And that was a major advance in science, and the understanding the structure of the genetic code. And weaved into this story is a woman that was not really acknowledged in a way that we would currently acknowledge people in science. And interestingly, what Jennifer took away from this, which I think was a really positive aspect of the way Walter perceived this situation, is that Jennifer, when she read the book, she was so excited by the science, and just the mention of a woman doing science in the book really propelled her to get on a plane and go to California and do undergrad in the and leave Hawaii for a career in science. And then the book really then starts to switch into a pretty heavy and technical, kind of very rapid foray into trying to get to the current day CRISPR technology. And this is where some folks might get lost. I think Walter did a relatively good job. He left out a lot of the other intricacies around how genome editing actually works, and I'm not sure if they were necessary, or but maybe more time could have been spent there. And so he starts back with Mendel and the genetic code and Darwin, and then rapidly kind of transitions through what DNA is, the Human Genome Project, and how sequencing the whole genome we thought would open up all these questions, and it did, but it answered a lot less than we expected, because a code's a code until you can decode it. And then it talks about Jennifer's early days as a scientist, how rather than working on DNA, which everyone was at the time, she was more interested in this cousin of DNA, RNA, and how her kind of inquisitiveness and taking a very, very risky route in terms of the project she was working on actually led to some pretty fundamental discoveries, highlighting that this kind of dogged curiosity and risk in science is important for those next breakthroughs. And so there's a clip that Walter talks about where Jennifer is interviewed on TV about one of her breakthroughs. So she was a well-known scientist even at a much younger age than she was currently. So already well-known in the area of RNA science. And so she was interviewed by TV, and she said that her technology might be able to be used for genome editing. And so the idea was already planted some 20 years before the discovery of CRISPR. And then from there, we switch into pretty hardcore kind of understanding of how the CRISPR systems were first hypothesized and then shown to be bacterial immune systems. And I'm going to talk a little bit about that and make it a little bit didactic, so hopefully I can inspire you to read the book with a little bit of pre-training. I don't know how well I'll go. The screen is a little far back, but and then we can skip really to the present, where we're actually starting to see some extremely promising clinical trial results for this technology. And then we'll finish it with some kind of more provocative and important discussions around bioethics and how that area is still very murky around this technology. And so I'm not sure if everyone can see this, but this is a bit of the genetic code from E. coli. And so this is a small bacteria that's in our guts. It's everywhere around us. And folks noticed that there were these repetitive bits of DNA in the locus. So this is in yellow here, in the genetic code. And there was some and before they even really understood what they meant, they came up with a hyper-technical name known as Crispr. So clustered regularly interspaced short palindromic repeats. That's just describing the nature of this repeat locus. And so often people would get really excited about repetitive sequences, but one scientist in Spain, Francisco Mohica, who used to work on bacteria that he finds in high salt pools on the ocean, and trying to understand how they can survive that high salt, was actually more interested in the sequences in between. And you can take these sequences and basically ask a database what those sequences might match to. It's like doing a Google search, effectively. And what he found is they match viral DNA. And so that was really odd. Usually you don't see viral DNA within the genetic code of bacteria. That's a little bit of a simplification, but in this kind of arrangement at least. And so the hypothesis, which was extremely profound at the time, and Walter does a good job of walking through, this guy, we considered Francisco Mohica this kind of lost hero or discoverer of CRISPR, because he doesn't get talked about enough, and hadn't been for many years, but now with these books have been written, some of the kind of unsung heroes of this technology are now being talked about more. So he had this hypothesis that it might act as a vaccination card, much like we do when we have a vaccine where we have either a dead virus or an mRNA encoding a bit of a virus. Our body sees it, it reacts to it, and says, oh, that doesn't look like us, so we'll mount an immune response. His idea was that these pieces of DNA were doing the exact same thing. And the reason that bacteria need an immune system is that much like there are viruses like COVID or Zika or HIV, there are bacterial viruses. And these are actually much more prevalent in nature than we'd expect. There's probably, I'm just throwing out a number here, about 100 million in our guts. And they're a big part of our ecosystem and the most abundant biological entity on the planet. So really fascinating. We work on these in the lab at Rochester. And in order for bacteria to survive, they have to mount an immune response against these viruses. Otherwise, the viruses will replicate and infect the bacteria and burst them open. And one interesting tidbit of information is that every day in the ocean, where there are tons of bacteria, 40% of bacteria are killed by phage. So there's this ongoing arms race of immunity and death between viral and host, much like we're seeing with the current pandemic. But this happens at the most kind of simple part of life. So these viruses have been around for billions of years. And so the idea was that maybe these CRISPR systems can protect against the viruses. And this is where the story gets really interesting. It's quite a great story to tell because these discoveries came from unexpected places. And Walter does a really good job of kind of teasing out all of these discoveries and putting it in the broad context of genomic editing. So long story short, Francisco had this great idea that was a hypothesis. So it hadn't been experimentally tested. A yogurt company that was interested in making their yogurt bacteria, so you need bacteria to make yogurt, Streptococcus thermophilus, make them immune to these bacterial viruses. Because in industry, if you have a viral infection in your yogurt plant, your production crashes, you lose hundreds of millions of dollars in yogurt, and you have to bleach the whole place and start again. And if you could engineer strains of bacteria that are resistant, and we have natural strains that are resistant, but if you can speed up that process, you can potentially prevent massive business losses. And so there was a research arm within this yogurt company called Danesco, now DuPont, that basically proved that this CRISPR system was an immune system, and they were able to effectively naturally immunize these bacteria. So there was no synthetic engineering going on. It was all using natural approaches to make immune bacteria. And so it's much like a vaccination card for these bacteria. And a lot of other research, dozens of labs across the world, are able to show that the way that this works is that the CRISPR-Cas system can detect the foreign DNA, or even some cases RNA, and then cut the RNA or DNA, and cutting that destroys the virus, because the virus generally doesn't have ways to repair its DNA, unlike us, where we have mechanisms to repair. So you cut the DNA in the virus and it dies. And so we consider them molecular scissors, and this is where the genome editing kind of idea comes from. And so this is where Walter switches and really gets involved in the race for to get this genome, this idea of using CRISPR in human cells so we can do genome editing. And this is where a lot of the interesting personalities come out in the book, the rivalries, the money. It kind of reads like a bit of a thriller in that sense. I mean, I'm biased. I was very embedded in that at the time, and so I'd be curious on what other folks thought when they read it, because we all kind of were interacting. And so can we program it to cut human DNA and do DNA editing? And so just as a quick timeline, and this is really the race that he captures in the book quite well, and the different personalities and that dogged competitiveness, and it's almost an East Coast versus West Coast kind of thing that was going on. And so you could write a rap song about that or something. But it very rapidly went from this initial paper in 2012. This is basically the manuscript that described the ability to take that CRISPR system out of bacteria and reduce it to the minimal components required and be able to cut DNA in a test tube. And that was what was effectively awarded the Nobel Prize, was what did we actually need to do the editing? And then what came quickly after were several groups, including Jennifer's group, that were able to put it in human cells and show that it can edit DNA in ways that weren't possible before. And then very rapidly, because this technology is so easy to use, it was used across pretty much all known organisms, and there are now human clinical trials, which I'll talk about in a minute. So not only in model organisms that we use in research, but also particularly important industrial crops. And there's an argument that maybe the biggest impact for CRISPR will be actually in agriculture and future-proofing our crops from climate change. And so there's a large amount of research in that area, and Walter goes into it a little bit. I would have liked to see a little bit more of that, because I think that's going to be powerful. It's much safer than the current ways of creating transgenic agricultural crops, and arguably considered not even transgenic, because you're not adding any extra genes, you're just changing the current ones. And so we really see the power of this is the idea that there are differences in the genetic code of different organisms or of humans that have disease, and we can change them back into something that is healthy. And so, for example, we have a bit of genetic code here, and there's a single change here that causes a disease. If we can take this and return it back to what it used to be, we might be able to treat that disease. And I have some examples here. So probably the most exciting example that we actually have clinical data on now is sickle cell. So sickle cell anemia, it's debilitating, very painful disease, and if left untreated, you die. And there are a bunch of other diseases, including cystic fibrosis and Fragile X, that can potentially be cured with this. And what Walter does, he starts talking about how you have this powerful technology, and naturally the universities want to turn that into revenue, and then there's a lot of money for potential therapy in this. And so he talks about how these different companies were formed around not only Jennifer, but also all the East Coast folks, and how there were rivalries, and how some of them dissolved and people left boards, and it all got very, very kind of juicy, and it was really well captured in this, and even things that I didn't know about, Walter was really able to pull out of Jennifer, because sometimes she was quite tight-lipped about this, but it's all written down here now in public knowledge. So I found that interesting. And they're going after a range of diseases. A lot of power in cancer immunotherapy, as you might be aware. Being able to supercharge our own immune system to target cancer, and there's some tricks that Crispr can help with that. Various congenital eye diseases, sickle cell, as I mentioned, and a range of other things that are actually currently undergoing clinical trials. And so I'll just quickly tell you about one case that Walter talks about quite nicely in here. And this is a little outdated already, but there's there's several patients, but I'm just going to focus on one, that use they've used genome editing to fix, in a roundabout way, the sickle cell bad hemoglobin. And what that causes is the red blood cells to end up long and sickled-like, and really what the danger is they clog up your arteries and end up in what's called these vaso-occlusive episodes. And so without going into the technology, because the way they did it's relatively roundabout and complicated, but the most heartening thing about this is, for this patient, it worked. And it worked so well that 17 months after treatment, and there's a figure here to show kind of how this is played out. She had to do a lot of transfusions, and she had a lot of these very painful vaso-occlusive events that would keep her in hospital for weeks. After she had the treatment, basically disappeared. So functionally cured. No hospital stays, no blood transfusions. And so Walter does a good job of talking about that. I would have liked a little more detail, but a lot of this data is actually even more recent than this book. Now, what Walter does after talking through all the technology and all the promises is there's a massive, massive bioethical debate about how to use this technology and the idea of recoding the human race, or the human species, as it should be properly said. And so as soon as this technology looked like it was going to work very well in humans and other organisms, people started to think about these questions. And Jennifer was very proactive about starting international summits where we bring in all the stakeholders, whether it's patients, bioethicists, scientists, religious communities, anyone that kind of has an interest in the power and the peril of the technology. And so Walter does a really good job of really digging into the development of that, and kind of looks at some of the key aspects, whether it's personal freedom versus what is natural, playing God. All these things get thrown around. The idea of equity issues, because this technology is extremely expensive to implement, $1 million a dose kind of regime. And so how can we make it more equitable for folks? And are there concerns about the haves and the have-nots with these kind of technologies? And so initially, and thankfully, scientists were pretty cautious. And so there's two ways that we can do, and I think I go into this after. I'll hold that thought for a moment. And so there are multiple international summits that really started to talk about this, and this is still ongoing. And depending on the school of thought, there are very different attitudes to whether we should progress with this versus whether we should not use it in cases where you could cure disease. And much like the debates around nuclear power or climate change or vaccine mandates, there's a lot of different stakeholders that have different opinions about how they should be used. And so there's basically this treatment versus enhancement dichotomy. When does a treatment become an enhancement? And Walter does an interesting job in one of the latter chapters where he talks about these things called thought experiments. And so one of the interesting thought experiments is that sickle cell is painful and horrible, but if you only carry one copy of the sickle cell mutation, you don't get a lot of disease, but you become effectively immune to malaria. And so in sub-Saharan Africa, this variant has actually increased in its proportion of the population because it has a positive selective effect. And so if we were to cure it, if we didn't want to pass, say I was half sickle cell and my wife was half sickle cell, and then our child would have it, would it be better to screen the embryos and look for an embryo that didn't have 2 of the copies and pass down that half sickle cell variant in case we encounter malaria, and that provides protection? And so there's this balance between fixing one problem but creating another and unintended consequences. And then he goes into some more interesting ones, which I found a little more difficult to digest, like the idea that because Miles Davis had sickle cell and he was taking a lot of drugs and alcohol to help him with that he would have never made the Creative Genius albums without being under the influence of both pain and drugs and alcohol, which is an interesting bioethical discussion, but it kind of gets a little murky. Or the idea that FDR wouldn't have been the great president he was if he hadn't had polio. And so there's a bit of discussion about that, and I found that fascinating, but a little tenuous. But it is apparently something that comes up in the bioethical field all the time about these unintended consequences. And also this idea of what is a disability, and do we want to cure deafness, or if there are if there's a family of deaf, a couple, and they want to have a deaf child because that's their world that they live in, who's to say that they can't do that or not? And so it gets a little scrambled in those later sections, and as a scientist, I actually don't feel qualified enough to fully make claims on that because it's really the regime of bioethicists that think in very different ways than we do. But I think he does an okay job. I think it's compelling and provocative, but I think one needs to be careful. And also the idea of individualism, consent, regulation, all of that. Similar argument around like community needs versus individual needs, and we've seen a wide variety of that lately in our current environment. But I think the take-home from all of that, and Walter really captures that right at the end, is that we need everyone involved to make these decisions. All the stakeholders need to be at the table discussing this, and perhaps we discuss it on an individual base-by-base cases. Everyone's got their different moral compass, and what might feel appropriate for one person may not be appropriate for someone else. And so back to the kind of didactic part of this, I wanted just to highlight for those that may not be aware, there are kind of 2 ways we can do genome editing. Say if I had a liver disease that needed to be treated, I could be injected with Crispr, it would potentially treat my liver disease, and that would be great. But I won't pass that on to any children that I have. However, if I was treated with for that liver disease at the single embryo cell stage, where I could pass down that genetic change in my gametes, so my germ cells, the sperm or the ovaries, then that would get passed down. And so the big discussion is really about the passing down of genetic mutation through future populations. The treatment of me as an adult, people are less worried about. And I think we'll see more of that before we see the germline editing. Another caveat is that germline editing is actually really hard to do and not that successful. And so there are certain kind of points that I probably just all really got across, but I don't know if everyone heard about the crazy news that came out of China a few years ago. The rule on the street basically was no germline editing. We're not going to do that yet. The world had decided we're going to hold up until the technology's there, because the consensus was it's not quite specific or good enough to get there. But there was a rogue Chinese scientist that basically went ahead and did it for all the wrong reasons, and he did it very poorly. And so this created quite a lot of upset in the scientific community, actually. I remember the news coming out and having to give a lecture that morning on this current stuff and being really, really kind of incensed. And did it for kind of reasons of fame and notoriety. He's now in jail. China put him in jail, and we don't know what's ever going to happen to him. I don't think we'll ever see him again. And he was a well-known scientist. He'd walked around in our circles before. And so after that shockwave happened at a genome editing summit. He came in and showed everyone the data, and everyone just kind of lost their minds. And so Now there's more and more talk about this. We knew that the cat was going to get out of the bag at some point, but it was a shame that it was done so terribly and just for the wrong reasons, that it was really irresponsible. So again, like many of our technologies, there are always these renegades that run ahead of the pack, and we have to try and keep them in check. I don't know how I'm doing with time. Am I way over time? Good. And so thinking about what the positive needs for germline editing are, so editing of the future generations. And so one way we can screen for disease is using pre-implantation genetic screening. So we look at the embryos, or we look at the fertilized eggs, and we see if we can do some very quick tests to see if they have the disease phenotype, the genetic disease or not. But sometimes that can't help pick embryos that will be disease-free. If it's two sickle cell patients having a baby, for example, is there a moral imperative for us to fix any future generations so that those parents don't have sickle cell in their lineage in the future, so their children don't have sickle cell? Or if you have a disease like Huntington's, that it's a dominant disease and it just gets passed on to everyone in the family effectively at a very high frequency. And so some of those, particularly where they're very low odds of getting a healthy embryo, we think that that's where the germline editing needs are. And then you start to get closer to that line. What about genes that can help you be less prone to having high cholesterol? There are certainly environmental ways of doing it, but what if we could actually change our genome to make sure that we could eat a little more fatty food and be okay? And there are certainly ways to do that. And then when you start getting over the line, it's like install mutations that offer a genetic enhancement. Say you want to be the fastest runner on the school team, and your parents aren't fast runners. And there is natural genetic variance. So if we test our athletes, a lot of them have genetic mutations that make them faster runners. We know that. And so is it equitable to give that to everyone so we can all be fast runners? Or do we play the genetic lottery? And so these are the questions that bioethicists are kind of toying with. And so where Walter kind of finishes this is with all of these kind of questions about morally, where does this all stand? Is it wrong to play God and interfere with nature? What is natural versus unnatural? And that line to many of us is very blurry, given how we understand biology. Does it jeopardize human dignity by altering the heritage of humanity? For future generations, there's no informed consent. You're passing this on to future generations without their consent. Is that an issue? Or will they come back and say, why didn't you edit me for sickle cell? And now I'm living with the disease. So there's that double-edged sword that's very hard to kind of tease apart. There's this problem with ableism and the identity of disability and what is a disability, and could that exacerbate discrimination? And a lot of other genetic inequities between the haves and have-nots, the fast runners versus the slow runners, and how do we tease that apart? And a big counterpoint to this that I want to get across is that unethical not to use germline editing to alleviate human suffering. If we could wipe out sickle cell tomorrow, is it unethical not to do that? And so this is kind of way above my pay grade, and this is why we need a big community effort to talk about this. And I don't think this is going to occur, but this is what gets talked about, the idea of eugenics, and I don't really want to touch that. And so to finish, I have a long list of things that I thought Walter really did well in the book. I think the descriptions of the technology and the discovery were very clear, and they were faithful. The unique personalities, ambitions, and quirks, I think he did an excellent job of. There are several other books in the same genre, and Walter has this ability to really tease out how people are really feeling. And there are things in here that I didn't know that he managed to tease out of my old boss. So I thought that was pretty cool. But he kind of put it in a way that was a positive spin on the competitiveness in science. He tackled the ethical questions pretty well, I thought. I think that's always a touchy subject, particularly for folks that don't think about it all day. I really liked the behind-the-scenes access to how the science worked. I think that was pretty spot on. And kind of what I think is the thrilling process of discovery and being a scientist. And I kind of, it echoes the feelings that I have as a scientist, so he did a good job with that. Criticisms, maybe the science is too technical. I'd love to hear your thoughts on that and what could be done. As scientists, we're always trying to improve our ability to communicate to the public. I thought it was overly long, and then there were some sections that were a little off topic. As I said, the ethical reasons can get very murky, but it's a murky subject by nature. And there's one big thing that I thought was missing is the limitations of the current technology. It's heralded as this like game-changing technology, which it is, but it doesn't mean it's perfect. And that imperfection's actually a big area of research right now, and it's quite a concern for things like germline editing. It's the discussion's more like whether we can do it, it's how good we can do it, and whether we don't make many mistakes along the way, because we can cause cancer with this as well if we don't do it right. And so I'd like to hear from everyone what your thoughts are on it that have read it, so I'm happy to take questions. Yes?